The Science Success Center, with funding from Title V, presents The Molecules of Cells, a biology workshop. Hi, I'm Daniel. Life's molecular diversity is based on the properties of carbon. Almost all of the molecules found in a cell are atoms of carbon bounded to one another and to other atoms. Organic compounds are carbon-based molecules that vary in carbon length. A carbon skeleton may be branched or unbranched. Sometimes carbon atoms may have double bonds as well. Carbon skeletons will sometimes form rings, which provide a more stable structure. Lastly, isomers are compounds with the same formula but have different structures. Characteristic chemical groups help determine the properties of organic compounds. The unique properties of an organic compound depend on the size and shape of its carbon skeleton and on the groups of atoms that are attached to the skeleton. Functional groups affect a molecule's function by participating in chemical reactions in characteristic ways. These groups are polar because their oxygen or nitrogen atoms exert a strong pull on shared electrons. The first functional group that we will discuss is the hydroxyl group, which consists of one hydrogen and one oxygen atom. This group will commonly form alcohols. A carbonyl group is composed of one carbon atom and one double-bonded oxygen atom. This group may be found at the ends of molecules, known as aldehydes, but when a carbonyl group is found near the center, it is known as a ketone. Next is the carboxyl group that consists of one carbon atom that has an oxygen double bonded to it and a hydroxyl group on the other end. Now we have the amino group. This functional group has one nitrogen with two hydrogens on each end. A phosphate group has a phosphate atom in the middle with four oxygen atoms bounded to that phosphate. The phosphate groups are highly important in the energy ATP molecules. Lastly, a methyl group is a carbon with three hydrogens bounded to the carbon. Cells make a huge number of large molecules from a small set of small molecules. This short polymer has three monomers and will continue to grow by adding specific monomers, so that the growing polymer becomes an important and specific molecule in the cell's body. Making polymers by a dehydration reaction. A dehydration reaction is a process that links short polymers with monomers. Keep an eye on the hydroxyl group attached to the short polymer end and the hydrogen attached to the monomer. Both the hydroxyl and hydrogen groups will detach themselves from their respective links and combine to make water. While the water molecule is being made, the monomer and short polymer will combine to each other, making a longer polymer. Breaking polymers by a hydrolysis reaction. A hydrolysis reaction uses one water molecule to break a link on a longer polymer into a shorter polymer and a monomer. Recall that in any reaction, no molecules are lost. Therefore, the hydroxyl and hydrogen groups will attach themselves to the ends of the individual monomer and the shorter polymer where the link was broken. Four major molecules are found in great quantities in all organisms. First are carbohydrates, which is our main source of energy to sustain life and perform activities. Lipids, also known as fats, are used for both energy and a source of materials to create a barrier used for separating different environments. Third are proteins. They are known for their unique three-dimensional structure and are important to the structures of cells and organisms and participate in everything they do. Last are nucleic acids, which are responsible for containing every living thing's genomic information. Next, we will talk more in depth about the four major molecules. Monosaccharides are the simplest carbohydrates. The glucose and fructose structures show the two trademarks of a sugar. A number of carbonyl groups and hydroxyl groups are found on all monosaccharides. Recall that when a carbonyl group is found near the center of a monosaccharide, it is a ketone. But when it is found at the end, it is more likely to be known as an aldose. Cells link two single sugars to form disaccharides. Here we have two single sugars of glucose, and by a dehydration reaction, the two monosaccharides link together to form a maltose, which is a disaccharide. The prefix mono means one, and di means two. Polysaccharides are long chains of sugar units. Polysaccharides are polymers linked together by dehydration reactions. These long chains may function as storage molecules or as structural compounds. Starch, a storage polysaccharide in plants, consists entirely of glucose monomers. Animals store excess sugar in form of another glucose polysaccharide called glycogen. Most of our glycogen is stored as granules in our liver and muscle cells, which hydrolyze the glycogen to release glucose when it is needed. Cellulose, the most abundant organic compound on Earth, forms cable-like fibrils in the tough walls that enclose plant cells. Carefully compare the oxygen bridges highlighted in yellow between glucose monomers and starch, glycogen, and cellulose. Another structural polysaccharide, chitin, is used by insects and crustaceans to build their exoskeleton, the hard case enclosing the animal. Fats are lipids that are mostly energy storage molecules. Lipids have fatty acids and glycerol building blocks. By dehydration reaction, we can combine the glycerol and fatty acid to make a lipid. Two types of fatty acids exist. The first has a saturated characteristic, meaning that it has the maximum number of hydrogens and have only single bonds. The second type of fatty acid is an unsaturated fatty acid that has fewer hydrogens and forcing a double bond where the carbon is missing a hydrogen, which causes a slight kink in the fatty acid chain. Phospholipids and steroids are important lipids with a variety of functions. Phospholipids have a glycerol head and two fatty acids attached to the glycerol. These phospholipid structures are important for cells because they make up the cell membrane, which provides an internal environment for the cell by forming a phospholipid bilayer. The hydrophilic heads are only interested in staying in contact with water, but the hydrophobic tails have the opposite taste. The tails do not want to be in contact with water. Cholesterol, a steroid. Steroids are lipids whose carbon skeleton contains 4 fused rings. Cholesterol is a common component in animal cell membranes, and animal cells also use it as a starting material for making other steroids, including sex hormones. Anabolic steroids pose health risks. Anabolic steroids are synthetic variants of the male hormone testosterone. Overdosing may cause violent mood swings and deep depression. The liver may also be damaged, leading to cancer. Proteins are essential to the structures and functions of life. Structural proteins are found in hair and the fibers that make up connective tissues such as tendons and ligaments. Contractile proteins are found in muscle and help with movement. Defensive proteins are the antibodies of the immune system, helping us fight off any foreign molecules trying to invade our bodies. Signal proteins can be hormones that coordinate body activity. More proteins. Receptor proteins may be built into cell membranes and transmit signals into cells. Transport proteins, like hemoglobin, transport oxygen in the red blood cells. Storage proteins, like an egg's egg white, serves as a source of amino acids for the developing embryo. General structure of an amino acid. Protein diversity is based on differing arrangements of a common set of just 20 amino acids. Amino acids all have an amino group and a carboxyl group. The structure of the R group determines the specific properties of each of the 20 amino acids that are found in proteins. Peptide bond formation. Taking two amino acids and chemically bonding them together by a dehydration reaction forms a dipeptide. The carboxyl group and amino group of both amino acids donate a hydroxyl group and hydrogen atom, respectively, to form water and a peptide bond. Performing numerous reactions similar to this will make a long chain called a polypeptide chain. A protein's specific shape determines its function. The 3D shape is what determines its specific function. Their shapes enable them to recognize and attach to its molecular target. In a process called denaturization, polypeptide chains unravel, losing their specific shape and as a result their function. So what may cause a denaturing? Changes in salt concentrations and pH can denature many proteins, as can excessive heat. A protein's shape depends on 4 levels of structure. The primary structure shows the polypeptide chain with its individual amino acid sequence. The secondary structure. In the second level of protein structure, parts of the polypeptide coil or fold into local pattern. Coiling of a polypeptide chain results in a secondary structure called an alpha helix. A certain kind of folding leads to pleated sheets. The double helix and the pleated sheets are held together by hydrogen bonds. The tertiary structure refers to the overall three-dimensional shape of a polypeptide. The indentations and bulges arising from its particular arrangement of coils and folds give the polypeptide the specific shape that fits to its function. Many proteins consist of 2 or more polypeptide chains, or subunits. Such proteins have a quaternary structure, resulting from the association of the subunits. Linus Pauling contributed to our understanding of the chemistry of life. Pauling was studying biological molecules. In 1953, he and a colleague discovered how oxygen molecules attach to iron atoms of hemoglobin. He also discovered how an abnormal hemoglobin molecule causes sickle cell disease. And it was also Pauling who first described the 2 fundamental secondary structures of proteins, the alpha helix and the pleated sheets. Nucleic acids are information-rich polymers of nucleotides. This is an example of a nucleotide. Notice the phosphate group and sugar group on the left-hand side. These 2 structures never change and are found on all nucleotides making up DNA. But the nitrogenous base can be of 4 different types: adenine, thymine, cytosine, and guanine. The nitrogenous base is what determines the name and property of the nucleotide. Sugar phosphate backbone. The sugar phosphate backbone is the repeating sequence of sugar to phosphate repeated numerous times. In this figure, the sugar and phosphate are represented by the blue and yellow colors. DNA double helix. RNA usually consists of a single polynucleotide strand, but DNA is a double helix, in which 2 polynucleotides wrap around each other. The nitrogenous bases protrude from the 2 sugar-phosphate backbones into the center of the helix. When A pairs with T and G pairs with C, it is known as base pairing. The 2 strands are held together by hydrogen bonds between their based pairs. Thank you for watching, everyone. Come visit us at the SSC if you have more questions. Good luck on all your studies, and tune in for the next workshop.